Hello students. In our previous session, we were studying about the experiment that was commonly called as Meselson-Stahl's experiment. With this experiment, these two scientists, they tried to prove that replication always occurs in a semi-conservative manner. So I have already explained to you what are the different modes of replication that were understood, that were studied before, but the most acceptable theory about the replication is the semi-conservative theory of replication. So you know why it is called a semi-conservative theory? Because in this, one of the strands of the parent DNA will be conserved or it will be retained in the daughter DNA. So when the cell replicates its DNA in the S phase of the cell cycle, so then what happens is, so there are two strands of the DNA, one will go with the one DNA molecule of the daughter cell, and the another daughter cell which is produced also will have another strand of the parent DNA. That is why it is called semi-conservative replication, and it was very well proved by this experiment. I'll just glance this experiment in a very briefly, and then we can move on to the next aspect of the replication. See, in the yesterday's class I had told you, in this particular experiment the tools that were used are the NH4Cl medium. Okay, I had told you there were two nitrogen sources, there were two media, nutrient media, one with N15 NH4Cl. That means the nitrogen source that was present here was the heavy nitrogen source, right? It was a isotope of nitrogen, that is N15 isotope. Whereas in this case, normal nitrogen, that is N14, right? So this was the normal nitrogen source, this was the heavy nitrogen source. So these were the two nutrient mediums, and E. coli cultures were used, E. coli bacterial cultures were used, and also the cesium chloride gradient centrifugation technique was also used in this particular experiment. What did they do here? Not to get confused. See, this experiment can be very important for your board examination, and it is often asked for 5 marks. Note this point. Okay, here what was done was, the bacteria were made to grow for many generations. One generation of E. coli will be completed within 20 minutes, and many such generations were allowed to grow on the heavy nitrogen source medium, that is N15 H4Cl medium, right? And after growing it for many generations, they were transferred to the N14 H4Cl medium, and they were allowed to grow for one generation, or even if they were allowed to grow for many generations, they were checked. That means a batch of that culture was isolated after every 20 minutes. Andre 20 ನಿಮಿಷ ಆದ ತಕ್ಷಣ ಒಂದು ಬ್ಯಾಚ್ ಅನ್ನು ತಗೊಂಡ್ರು, and the DNA of that was considered for testing. So again they were left to grow for another 20 minutes, that is to complete one more life cycle, and after that is 20 minutes they took out one more batch of that culture, like that. So at every regular intervals they took out the cultures and they looked for the DNA. So when they looked for the DNA, that DNA was having the heavy strand, that means the strand which was having the heavy nitrogen, the other strand was also having the light nitrogen, that is N14 nitrogen. So this was how it was proved that the replication always occurs by semi-conservative manner. So these diagrams it can be well explained with the help of these diagrams, and you need to draw these diagrams in the examination. And see, it was not left there. The DNA extract from those cultures which were extracted at regular intervals from this particular medium, that DNA was crushed and it was made into solution, and it was subjected for centrifugation. I have already told you what is centrifugation. Centrifugation means you just have to take the sample in the test tubes and you have to put it with the centrifuge, and you have to rotate it at a very fast RPM, maybe 5,000, 6,000, maybe up to 10,000, 15,000 RPM, right? And when you do that, obviously the heavier particles, they settle down and the lighter particles come up. That is what will happen here. The CaCl was considered, cesium chloride was considered as the medium for centrifugation, because CaCl solution has got more density than that of the DNA, and obviously DNA will float in that medium. And when N15 was centrifugated, N15 formed a band somewhere near to the lower surface or lower bottom of the test tube. When only N14 DNA was considered, that formed a band little upper to that of the N15 one, right? And when this DNA, which was said to be having one strand of N14 and one strand of N15, was considered for this thing, they formed one band for N14 and N15. That means there was a single band combined by both N14 and N15, and there was another band that was only having N14. So this confirms what? The semi-conservative replication mechanism in the case of the eukaryotic, in the prokaryotic cells, that is in the E. coli cells. Now let us talk about the enzyme machinery and the process of replication. So as we were discussing about the replication, see, replication, it is well understood and it was first studied in the prokaryotic system, obviously with the help of the bacterium Escherichia coli, right? So in order to understand in the early stages, the replication was well studied in prokaryotes, and then it was also studied and understood in the eukaryotic cell system. So there is no much difference, but the complexity of the process will vary from prokaryotes to eukaryotes. Now let us try to understand the process of replication, but before we understand the process or mechanism of replication, we need to know what enzymes are participating in the process of replication and what role they play and how they contribute for the entire mechanism of replication. See, the main enzymes involved, one important, very important enzyme that is involved in the process is, that is called as DNA dependent DNA polymerase. DNA dependent DNA polymerase. Remember, ಇನ್ನೊಂದು enzyme ಬರುತ್ತೆ ನಿಮಗೆ while learning about the process of transcription, that is next, which is the next process after replication. There you will come across another enzyme called as DNA dependent RNA polymerase. But here in the process of replication, you are learning about DNA dependent DNA polymerase. Okay, so here DNA polymerase is the enzyme that is responsible for the synthesis of the new DNA. Replication, you already know, replication means what happens? So by having one guiding strand or template strand, a new strand of the DNA is produced during the S phase of cell cycle, yes or no? So in order to synthesize that new DNA strand, the enzyme that is required is DNA polymerase. It is called as DNA dependent DNA polymerase. Why? Because this will function completely based on complementarity based pairing. What does that mean? Here, look at this. This is the strand of the DNA. Okay, these are the two strands of the DNA. That is the parent DNA. Imagine there is some bases here, some sequence of the base here, A, G, C, T, C, T, A, C, T, G. Okay, and this should be complementary to this strand. So now when A is here, T should come here, C, G, A, G, A, T, G, A, C, right? So now what will happen? Both of these strands of the parent DNA will act to synthesize two new strands, right? So one new strand comes here and the other new strand comes here, and this strand will be having the sequence of the bases, so which will be complementary to this one. Illi T idre A barutte, illi G, C, T, right? So this strand will be exactly like that of this strand, yes or no? Try to understand. Nodi, ee eradu strand galu complementary agide, right? And this strand will synthesize this particular new strand, and this will help in the synthesis of this particular strand. And the synthesis of the new strand of the DNA is completely based on the complementarity based pairing, right? So illi T idre A barutte, C idre G barutte, G idre C barutte, A idre T bantu, G idre C bantu. Again A is there, T comes here, T is there, A comes here, G is there, C comes here, A is there, T comes here, C is there, G comes here. Now check for the sequence A, G, C, T, C, T, A, G, C, T, C, T, A, C, T, G. Same sequence as that of the parent strand. ಎಸ್ ಆರ್ ನೋ? ಹಾಗಾದ್ರೆ ಇಲ್ಲಿ ಬರುವಂತಹದ್ದು ಏನು ಬರುತ್ತೆ? ದಿಸ್ ಸೀಕ್ವೆನ್ಸ್, ದ ಸೀಕ್ವೆನ್ಸ್ ಆಫ್ ದಿಸ್ ನ್ಯೂ ಸ್ಟ್ರಾಂಡ್ ವಿಲ್ ಬಿ ಎಕ್ಸಾಕ್ಟ್ಲಿ ಲೈಕ್ ದಟ್ ಆಫ್ ದಿಸ್ ಪೇರೆಂಟ್ ಸ್ಟ್ರಾಂಡ್. ರೈಟ್? ಸೊ ಹಾಗಾದ್ರೆ ಇದೆರಡು ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಆಯಿತು, ಇದೆರಡು ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಆಯಿತು. ದಿಸ್ ವಿಲ್ ಫಾರ್ಮ್ ಒನ್ ನ್ಯೂ DNA molecule, this will form one new DNA molecule. So this is how the replication occurs, and this completely is based upon what? Complementarity base pairing, yes or no? And the synthesis of this new DNA will be done with the help of, or it will be catalyzed with the help of this particular enzyme, which is called DNA polymerase. Right? So it depends on what? The old DNA or parent DNA, that is why it is called DNA dependent DNA polymerase enzyme. And this, while learning about the DNA dependent DNA polymerase enzyme, we will come across certain characteristics, certain important characteristic features of this particular enzyme that we need to understand. First feature is that here it is highly efficient. Highly efficient. The DNA dependent DNA polymerase is highly efficient and also highly specific. Highly efficient and highly specific. How do we justify these two things with respect to the DNA dependent DNA polymerase? Let us look into that now. See, highly efficient means here this DNA dependent DNA polymerase works very rapidly, but in a very confined way. Okay, so even though it works very fast, it will be very specific. How specific? It will be having, always it follows the complementarity base pairing, follows complementarity base pairing, complementary base pairing, and the DNA dependent DNA polymerase always will be functioning in only one direction. This functions in only one direction, that is 5 prime to 3 prime. Remember, right? E directionally matra aguvantadidu, right? And how rapidly it happens? For this, we need to consider an instance. Like for example, in E. coli, there are 4.6 into 10 raised to 6 base pairs. In the DNA of the E. coli, there are 4.6 into 10 raised to 6 base pairs, right? So, the entire DNA of the E. coli will be replicated in 38 minutes. In 38 minutes. The entire DNA of the E. coli will be replicated in 38 minutes, which accounts for approximately 2000 base pairs per second. Okay, see, now you can also calculate it for human, right? So in human DNA, in the haploid content of the human DNA, that the amount of the base pairs is 6.6 into 10 raised to 9 base pairs. Okay, so if that is the amount of the DNA, you can calculate. So this was a question in NEET examination, so you note that point and try to calculate. Okay, maybe in the CET class I would like to tell you about that, right? So if this is the time consumed to replicate entire DNA of E. coli, which is of the length that is 4.6 into 10 raised to 6 base pairs, so it will take approximately per second it will replicate 2000 base pairs. Okay, that is a very fast process, right? It's a very rapid, it's a highly efficient process, right? And it is also highly specific because it always follows complementarity base pairing, and also it happens always in only a single direction, that is 5 prime to 3 prime direction, right? And any mistakes, if there are any mistakes are there, then so this polymerase will also help in correcting those mistakes, and it also has the capacity to stop, right, the process of replication there. Now the other enzymes that are involved here, see, this is one particular enzyme that is a DNA dependent DNA polymerase. The other enzymes such as ligase, helicase, okay, so these are the other enzymes, and also which one, primase, Okay, these are the other enzymes. So these are not being mentioned in the textbook, but still for your knowledge, you need to know about this. Ligase enzyme will help in joining small fragments of DNA. Okay, the small fragments of the DNA. Imagine there are many small fragments of the DNA here. All of them they are joined together with the help of the enzyme called DNA ligase, or simply ligase enzyme. Then helicase enzyme. See here, while replication happens, you know that the structure of the DNA will be a double helical structure like this. So before the replication process starts, this helical structure has to be unwound. Adu open agbeku. It has to be unwinded. It has to be opened, and the open arms will act as the template strands. Template means what? See, this strand will be helping in the synthesis of a new DNA strand. So this will become template to this new DNA strand. Similarly, this strand also will help in the synthesis of this new strand. Now this strand will become template to this strand. So template strand. So in order to have open template strands, the unwinding of the helix has to happen. Unwinding means opening up of the helix has to happen. So unwinding will be catalyzed by the enzyme called helicase enzyme, right? And primase. See, in the process of replication, while we learn about the process of replication, there are certain small fragments of the RNA. They are called as RNA primers will come into picture. RNA primers will come into picture. So synthesis of RNA primers will be catalyzed by the enzyme called primase. Okay, so just for your knowledge, you need to remember these things. So it is very important for you to learn about the enzymes and understand their functioning ability, so that you will be able to better understand the process or mechanism of replication, which we are going to study now. So now let us study the process or mechanism of replication. Mechanism of replication. For this mechanism of replication, we have already understood the requirements. I'll just write the requirements here. See, requirements for the process of replication are the enzymes. So we have already listed which are the enzymes required. That is the DNA dependent DNA polymerase, which will be functioning very efficiently in a single direction and also very accurately on the basis of complementarity base pairing, right? And if any mistakes are there, it has the capacity to stop. And if that particular enzyme, DNA dependent DNA polymerase, goes on replicating, then it will lead to mutations. Okay, that is one enzyme, and other various enzymes are also involved, just like how we have listed out ligase, helicase, and also the primase, and many such enzymes are also involved in this, right? So this much detail is enough for your level now, right? And after that, there are also certain molecules which will be acting as dual functions, like DNTPs. Okay, see, deoxyribonuclease triphosphates. Okay, so these DNTPs, they act as both substrate molecules to produce the new DNA strand, and also they will act as the energy molecules. That means this replication is a very high energy consuming process. It requires a lot of energy to unwind the DNA, to produce the new strands of the DNA, right? And because of the high accuracy and because of the high efficiency of the DNA dependent DNA polymerase enzyme, it will require a lot of energy in order to do all these functions in the cell. In order to carry out with the supplement of all that energy, so these molecules will act as the energy molecules as well, along with acting as the substrate molecules to synthesize the new DNA. So ATP molecules, DATP, GTP, okay, MTP, so all those molecules will be acting as energy molecules here, right? And so the mechanism of the DNA replication, see, you need to understand, replication originates not rapidly at any point, at one particular point called origin of replication. Okay, See, if this is the DNA molecule, if this is the DNA double helix structure of the DNA, replication will not just start at any point on the DNA. Replication will start only at that point which can be recognized by the set of enzymes. You know which are the set of enzymes. ಯಾವುದೆಲ್ಲ enzyme ಇರುತ್ತೆ? Helicase. Try to remember. Helicase ಏನು ಮಾಡುತ್ತೆ? Helicase will go and bind to one particular point, and one particular point ಅಂತ ಹೇಳಿದ್ರೆ a particular sequence. Okay, if this is a DNA double helix structure ಅಂತ ಹೇಳಿದ್ರೆ there is some sequence, say for example CGT, CGATCG, some sequence is there here, right? And complementary base ಕೆಳಗಡೆ ಇರುತ್ತೆ. So ಇದೇ particular sequence ಅನ್ನ ಅಥವಾ ಇಂಥದ್ದೇ ಮತ್ತು ಯಾವುದೋ sequence ಅನ್ನ I have only taken random example here. So this will be recognized by a set of enzymes, and from there it will start unwinding, okay, and then the process of replication will start. So that point or that small stretch on the DNA with a particular sequence, right? That will be called as origin of replication. It is also referred to as ori anta kariteve in some prokaryotes, or in some eukaryotes it will be also referred to as oric point anta kuda kariteve, oric point or oric point anta kuda kariteve, right? So that is why, see, in the biotechnology chapter you are going to learn, see, why some small fragment of the DNA from any random cell, say for example there is plant X, ಒಂದು coconut plant ಇದೆ ಅಂತ ಅನ್ಕೊಳ್ಳಿ. So if you isolate the DNA from one of the cells of the coconut plant, ಒಂದು ಸೆಲ್ ನಿಂದ ಸ್ವಲ್ಪ DNA ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಮಾಡಿಕೊಂಡು ಅದನ್ನ ನೀವು ಯಾವುದೋ ಇನ್ನೊಂದು ಸೆಲ್ಗೆ ಹಾಕಿದ್ರಿ ಅಂತ ಹೇಳಿದ್ರೆ ಆ DNA ಅಲ್ಲಿ ರೆಪ್ಲಿಕೇಶನ್ ಆಗ್ಲಿಕ್ಕೆ ಆಗುದಿಲ್ಲ. The plant DNA which you have put in some animal DNA ಅಂತ ಅನ್ಕೊಳ್ಳಿ, ಆಗ ಅಲ್ಲಿ ರೆಪ್ಲಿಕೇಶನ್ ಆಗ್ಲಿಕ್ಕೆ ಚಾನ್ಸ್ ಇಲ್ಲ. It will just degenerate. That DNA will be dead. It will degenerate. Why does that happen so? Because ಈಗ ನೀವು ಸ್ಮಾಲ್ ಸ್ಟ್ರೆಚ್ ಆಫ್ DNA ಕನ್ಸಿಡರ್ ಮಾಡಿದ್ರಿ, ಅದರಲ್ಲಿ ಪಾಯಿಂಟ್ ಆಫ್ ಒರಿಜಿನ್ ಆಫ್ ರೆಪ್ಲಿಕೇಶನ್ ಇಲ್ಲ ಅಂತ ಹೇಳಿದ್ರೆ ಅದು ರೆಪ್ಲಿಕೇಟ್ ಆಗೋದೇ ಇಲ್ಲ. ಬಿಕಾಸ್ ಇನ್ ದಟ್ ಸ್ಮಾಲ್ DNA fragment, when there is no particular sequence to be identified by the set of enzymes, then how can it get replicated? And that is why we make use of vectors. We make use of vectors which will be having the point of origin for replication, and point of origin of replication ಇದೆ ಅಂತ ಹೇಳಿದಾಗ, so ಇಲ್ಲಿ ori point ಇದೆ ಅಂತ ಅನ್ಕೊಳ್ಳಿ, right? And here you are going to cut a small fragment of that vector, and you are going to insert your interest of DNA of your interest, and then now this vector will be placed in the host cell. ನೀವು ಯಾವ್ದು ಸೆಲ್ ಇಡಬೇಕು ಅನ್ಕೊಂಡಿದ್ರಿ ಆಗ ಇಟ್ಟಾಗ, so this will be recognized by the set of enzymes, and this will be replicated along with that DNA which is inserted, the foreign DNA which is inserted, right? So that is the importance of point of origin of replication. Now at that point of origin of replication, say for example ಇಲ್ಲಿ point of origin of replication ಇದೆ ಅಂತ ಅಂದ್ಕೊಳ್ಳಿ, and then so enzymes they will identify that particular point, and slowly unwinding of the helix takes place. Here, see here, unwinding of the helix takes place like this. So origin point and recognize ಮಾಡಿದ ತಕ್ಷಣ unwinding takes place. Unwinding leads to the formation of Y fork, or it's also called Y junction ಅಂತ ಕೂಡ ಕರಿಬಹುದು. It is called Y fork or Y junction. So unwinding of the DNA helix, you know, unwinding will be catalyzed by the enzyme DNA helicase, right? So that enzyme DNA helicase will open up both the sequence, both the strands of the DNA, and it will form long stretch of both the strands of the DNA, and this forms Y-shaped junction called Y junction or Y fork. Yes, so this is how it will be formed. And then what happens now? See here, the further process of the DNA, when DNA both the strands are opened up like this, you know, the two strands they are anti-parallel to each other, right? So see this strand, this is 5 prime, this is 3 prime, this is 3 prime, and this will be 5 prime, correct? No? So this you have already considered, we have already understood that why they are anti-parallel while studying about the structure of the DNA, right? So when this is like this, now which enzyme should come into picture? The enzyme DNA dependent DNA polymerase. DNA dependent DNA polymerase will come into picture for the synthesis of new strand of the DNA, and you know this will be functioning very accurately in only one direction, that is from 5 prime to 3 prime direction. ಅಂದ್ರೆ ಹೊಸ strand of the DNA production ಆಗಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ it can happen only in one direction, that is 5 prime to 3 prime. ಹಾಗಿದ್ರೆ ನೋಡಿ ಇಲ್ಲಿ 5 prime to 3 prime ಆಗಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ ನೋಡಿ it cannot happen in this way, because ಇಲ್ಲಿ 5 prime ಅಂತ ಹೇಳಿದ್ರೆ again 3 prime comes here, so 3 prime ಒಟ್ಟಿಗೆ parallel ಆಗಿರಲಿಕ್ಕೆ ಚಾನ್ಸ್ ಇಲ್ಲ, so it has to be anti parallel. Yes or no? Here also it has to be 5 prime to 3 prime, not like this, because 3 prime should come here. ಇಲ್ಲಿ ಸಿಂಥಸಿಸ್ ಆಗುವಂತಹ ಹೊಸ ಸ್ಟ್ರಾಂಡ್ ಇರೋ 3 prime ಇಲ್ಲಿ ಬರಬೇಕು and 5 prime ಇಲ್ಲಿ ಬರಬೇಕು, which is acting here. This is acting as template strand. So this template strand will help in the synthesis of a new DNA strand, which is whose 3 prime end should come here and 5 prime should come here. And so the new strand produced by this particular template strand should have its 5 prime end here and 3 prime end here. Yes, that means here one strand is being produced 5 prime to 3 prime in this direction, one strand is being produced 5 prime to 3 prime in this direction. That means replication takes place bidirectionally, though replication, the process of replication is bidirectional. ಯಾಕೆ ಅಂತ ನಿಮಗೆ ಗೊತ್ತಾಯ್ತು ಈಗ ಅಲ್ವಾ? Because one parent strand is 5 prime to 3 prime, the another parent strand is 3 prime to 5 prime. But DNA dependent DNA polymerase can function only in 5 prime to 3 prime direction. So ಹಾಗಾಗಿ ಇಲ್ಲಿಂದ 5 prime to 3 prime ಅಂತ ಹೇಳಿದ್ರೆ ಹೀಗೆ ಬರಬೇಕು. It is coming towards the point of origin of replication, whereas another is moving away from point of origin of replication. I hope you are understanding, right? So this particular strand which is coming towards the point of origin of replication, ನೋಡಿ ಇಲ್ಲಿಂದ ಹೊರಗಡೆಯಿಂದ ಒಳಗಡೆ ಡೈರೆಕ್ಟ್ ಆಗ್ತಿರುವಂತಹ ಅಥವಾ ಸಿಂಥಸಿಸ್ ಆಗ್ತಿರುವಂತಹ ಸ್ಟ್ರಾಂಡ್ಗೆ ನಾವು it is called leading strand ಅಂತ ಕರೀತೇವೆ. It is called leading strand. The other strand which is produced on the other parent template strand, which is moving away from the point of origin of replication, that will be called as what? It is called lagging strand anta kariteve. Lagging strand anta kariteve. Okay, see, lagging strand is always produced on the template strand which is having 3 prime to 5 prime direction. Yes or no? ನೋಡಿ ಒಂದ್ಸಲ 3 prime to 5 prime direction ಇರುವಂತಹ ಟೆಂಪ್ಲೇಟ್ ಅಂದ್ರೆ ಏನು ಅಂತ ನಿಮಗೆ ಕ್ಲಿಯರ್ ಆಗಿ ಗೊತ್ತಾಗಿದೆ ಅನ್ಕೊಳ್ತೇನೆ ನಾನು. ಟೆಂಪ್ಲೇಟ್ ಮೀನ್ಸ್ ಇದಕ್ಕೆ ಟೆಂಪ್ಲೇಟ್ ಅಂತ ಕರಿತೇವೆ ಅಲ್ವಾ? ಹೊಸ DNA ಅನ್ನ ಪ್ರೊಡಕ್ಷನ್ ಮಾಡಲಿಕ್ಕೆ ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಬೇಸ್ ಪೇರ್ಗಳನ್ನು ಒದಗಿಸ್ತಿರುವಂತಹ, ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಬೇಸ್ ಪೇರ್ಗಳನ್ನು ಕೊಡ್ತಾ ಇರುವಂತಹ, ಪ್ರೊವೈಡ್ ಮಾಡ್ತಿರುವಂತಹ ಸ್ಟ್ರಾಂಡ್ಗೆ ನಾವು ಏನದು ಟೆಂಪ್ಲೇಟ್ ಸ್ಟ್ರಾಂಡ್ ಅಂತ ಕರಿತೇವೆ. ಸೊ ಈ ಹೊಸ ಸ್ಟ್ರಾಂಡ್ಗೆ ಇದು ಟೆಂಪ್ಲೇಟ್ ಸ್ಟ್ರಾಂಡ್ ಆಯಿತು, ಈ ಹೊಸ ಸ್ಟ್ರಾಂಡ್ಗೆ ಇದು ಟೆಂಪ್ಲೇಟ್ ಸ್ಟ್ರಾಂಡ್ ಆಯಿತು. ಸೊ ಈಗ ಟೆಂಪ್ಲೇಟ್ ಸ್ಟ್ರಾಂಡಿನ ಪೊಲಾರಿಟಿ ಯಾವ ರೀತಿ ಇದೆ? ದಿಸ್ ಈಸ್ 3 prime to 5 prime. ಸೊ 3 prime to 5 prime ಇರುವಂತಹ ಪೊಲಾರಿಟಿಯನ್ನು ಹೊಂದಿರುವಂತಹ ಯಾವುದು ಟೆಂಪ್ಲೇಟ್ ಸ್ಟ್ರಾಂಡ್ನ ಮೇಲೆ ಪ್ರೊಡ್ಯೂಸ್ ಆಗ್ತಿರುವಂತಹ ಹೊಸ DNA ಸ್ಟ್ರಾಂಡ್ಗೆ ನಾವು ಲ್ಯಾಗಿಂಗ್ ಸ್ಟ್ರಾಂಡ್ ಅಂತ ಕರಿತೇವೆ. ಓಕೆ, whereas if the template strand has got the polarity that is 5 prime to 3 prime, if the template strand has got the polarity that is 5 prime to 3 prime, then it will be called as, this strand will be called as leading strand anta kariteve. The new DNA strand that is produced on that template strand will be called as leading strand, right? So this is one more point that is to be understood. But another complexity is there with respect to this one, especially with the lagging strand. See, on the lagging strand what happens? The entire DNA strand is not made at once. ಒಂದೇ ಸ್ಟ್ರೆಚ್ ಅಲ್ಲಿ ಆಗುವಂತದ್ದಲ್ಲ. You know what is leading strand, what is lagging strand, right? So the strand which is formed on the template which is having the polarity 3 prime to 5 prime, I repeat, the new strand of the DNA which is synthesized on the template which is having the polarity 3 prime to 5 prime will be called as, that new DNA strand will be called as lagging strand, and this lagging strand, it is discontinuously formed strand. So here it undergoes discontinuous replication. Why is that so? I mentioned about one more enzyme called primase, if you remember. Primase enzyme is responsible for the synthesis of small fragments of the RNA. Nodi, I'm going to draw one more similar diagram here. See, this is the template strand with here 5 prime and 3 prime. Okay, see, illi DNA dependent DNA polymerase will catalyze the synthesis of the leading strand in this direction. This is continuous replication on leading strand, whereas on the lagging strand what happens here, this strand, it is not continuously synthesized, but primase enzyme will help in the placement of smaller fragments like this. These are called as primers and these are called as primers. These primers are of RNA. These are called as RNA primers and these RNA primers at the end of, see here, this is the 3 prime end of the RNA primer, this is the 3 prime end of the RNA primer. This will provide a free OH group, right? And in between that illondu DNA bantu, and here once again there will be a DNA coming. So illi baruvantaha small DNA fragments enirutte, those small DNA fragments are later joined together to form one continuous strand. That is why small DNA fragments bandu adana later join ಮಾಡೋದು ಯಾರು ಅಂದ್ರೆ ligase. DNA ligase will act to bind those or stitch those small fragments of the DNA together to form one long stretch of the DNA. See, this is how the formation of the DNA takes place on the lagging strand, and that is why it is called discontinuous replication. Discontinuous replication on lagging strand. Remember, okay? See, one simple diagram that is as given in the textbook I'll try to draw here, and that diagram is also very important for you to draw in the examination. So this is all for your understanding. So that simple diagram as given in the textbook I'm going to draw here. See here, this is the Y fork that is formed because of the unwinding of the DNA helix. This is 5 prime and 3 prime end, so anti-parallel. This is 3 prime, this is 5 prime. Yes, so you know DNA has to be formed only in one direction, that is 5 prime to 3 prime. Now this will become the leading strand, right? And this will become the lagging strand. So this is discontinuous replication, whereas this one is continuous replication, right? And you should also not forget to mention the polarity of the newly synthesized strands. So this is 5 prime to 3 prime, this is 5 prime to 3 prime, like this. Okay, I hope you understood the process of replication. This is about the mechanism of replication. Okay, so enzymes and machinery of the replication process you have understood. It's a very important question for 5 marks. Practice the diagram and note down the points regarding this. If you don't understand anything, you can call me and ask doubts. Thank you.